Good morning, everyone. Welcome to episode number 14 now from the AWC webinar. Today we're going to be covering everything you need to know, or let's put just microplastics, or what you need to know about microplastics. Today we're joined by Dr. Nicole Fahrenfeld. She's the associate professor at the Rutgers University in New Jersey, or might be New York. So she's coming out to us from the East Coast. We're going to be covering some of that. For those of you that this is the first time, this is something that we're hosting every Thursday, 10 o'clock East Standard Time, 1 p.m. East Standard. I always kind of cover both. These are some of the topics that we've covered before. This week in October, we're doing emerging contaminants, so we're covering, this is now our second day. We've had to shuffle the dates around a little bit, so today we're doing microplastics. If this is your first, again, kind of a continuation to what we're going to be covering this month with all four of them. There's, of course, more of them, but this is part of the Water Week in October. We thought we could kind of feature the four ones that are kind of coming up the most in the news. For those of you that are not familiar with AWC, if you haven't been with us before, we design and manufacture water treatment plants. We have close to 600 both water and wastewater treatment plants worldwide now. That's not just in North America, but we're expanding into Latin America and basically everywhere. So some of the ones that we cover, of course, include potable water. We do wastewater and skid-mounted systems. When we look at microplastics in this particular case, it's going to be a very unique approach as to how we treat them. Today, we won't be going after the treatment itself, but more on the contaminants and where they come from, what they are, and what they can affect both both humans and any other ecosystems. So with that, we're going to bring it over to microplastics, and Nicole, take it away.
Okay, hopefully you can see my screen.
Yeah, we can see it. Go for it.
Great. Thanks for joining me today. I don't know if I'll tell you everything you could ever know about microplastics, but I'll certainly give you an intro to my understanding of fate and impacts, and along the way I'll sprinkle in some of the results that are from my lab. So we're talking about the same thing. Generally, when people are speaking about micro or nanoplastics, we're talking about man-made polymers that are less than 5 millimeters in diameter. The image on the right here is one that I borrowed from presentations from the Woods Hole Oceanographic Institute, and I like it because it's showing you sort of the expected increase in concentration as you get smaller for plastics. Here they estimate how much nanoplastic could come from one PET water bottle. And they also show on this size range what a lot of the research had focused on in terms of sampling is using these plankton nets. And you can see that that's only capturing a small amount of the plastic that could be potentially out there. And one other thing I really like about the image that they have here is that they're also showing you sort of a qualitative that you're increasing the potential for biological uptake from the intestine as you're moving to the left here, to the smaller sizes. So it's something to point out that we might be expecting more uptake, and with that perhaps impacts as you move to the left. And a lot of the work that folks are doing is focused on the right, partly because the techniques are not fully there for nanoplastics widespread. There are a variety of techniques certainly available in the meso and the microplastic size. So we usually divide microplastics into two classes to begin with. Primary, those that were manufactured in small sizes, like what I'm showing you here, a picture of what was probably added to some personal care products. It's rare that we observe those in the environment. At least my team has not observed them very often. Most of what we're observing are these secondary plastics, so something that was broken down from something larger, so maybe fibers or fragments and so on. If we wanted to think of a broad conceptual model for fate and transport once the plastics are released into the environment, I like this one from JGR. So another group put this together, basically showing that plastics that are in fresh water are eventually put into the ocean where they have circulation and can get into faraway places. But otherwise, we'd be expecting there to be weathering and breakdown so that we're moving towards those smaller size plastics. And also ingestion When one thing that we would think about there is that ingestion, biofouling, both could be helping lead to settling. So eventually sinking even of the buoyant plastics. So the one thought is that you're going to see eventually plastics moving towards the sediments. Although how long it takes to get there is certainly an interesting question to explore. And one other thing I really like about this image is that they're including atmospheric deposition. There's a growing interest in that, particularly when it comes to microfibers. It's really hard to keep your samples uncontaminated from microfibers because a lot of the clothing that you wear will be shedding those. And atmospheric samples have also shown things like tire from road wear. So I think that'll be an interesting area for further study. Some of the work that my team has done is focused on sampling surface water, moving from riverine environments out into estuaries. So here you're looking at, this is the Raritan River and the Hudson River, and sampling we did out in the Raritan Hudson Bay. So here we did focus on using some plankton nets, so you can see that we're limited in how small we could go. And we also did some source sampling, so wastewater, influent and effluent, and stormwater. And just to briefly explain, we did sampling anywhere from the ambient ocean out to the mouth of the river. So some examples of what we're seeing, again, it's mostly secondary plastics. Here's what a sample might look like on a mesh filter. And we use analytical techniques, mostly spectroscopic, FTIR, micro FTIR, FPA, FTIR, Raman spectroscopy, and so on for these. But a lot of these you could pick out, I think, with your eye pretty reasonably well, at least as a starting point. Like everything that's bright blue and bright yellow here was a plastic. You didn't realize I was giving you a test for the picture on the right. One of these wasn't a microplastic, and it's this that ended up showing up as wool. So if we look at some of the results that we've seen out in surface water systems from freshwater out into the ocean, and here focusing on the larger size class, you see one story, which is that the highest concentrations are present here at the mouth of the river. So consistently this low flow day, a low flow day, a high flow day, the highest concentrations on these bubble plots you're seeing there. I won't show you the data, but I'll just say the patterns are really different for smaller microplastics, which if this is something you're interested in studying and testing, it really begs the question as to what size class you're looking at. And if you're reviewing the literature, I think it's something to consider as well. Examples of the type of polymers that we observe. Here you can see from one of the sampling dates, it's really a lot of polyethylene. Some of that's biased to our methods because we're sampling the surface there and focusing on the buoyant plastics, and those are definitely buoyant plastics. Polypropylene, polyester, polystyrene, and so on. And then other particles that we're seeing that were non-plastics that are basically surviving the extraction techniques. So that does raise the question, these polymers, what are the sources of them? And one thought could be like, well, could we link the polymer type to where it came from, what it was initially used for? And this is a figure from a Plastics Europe report where they basically show how much overlap there is for the several different polymer types across different industries. So if you're just going on the major polymer type, it's probably not going to necessarily answer that question. Although if you can do more advanced work saying that these polymers, really it's not just polyethylene, it's a mixture of plasticizers and dyes and other things. Maybe if you could get that deep, you might have a better chance of backtracking it. We did a literature review of some different pathways of entry of plastics into the environment. Here, wastewater and compost were there. And here I just want to point out, it's a heat map showing you again, it's polyethylene, polypropylene, down here polyester. Those are the most commonly observed polymers across several studies. So I showed you some of our results from the Raritan Hudson Bay, and I wanted to put them into context. How does that compare to different pathways of entry or sources into the environment? So for that study, I said we also did some wastewater influent sampling, effluent sampling, as well as sampling. So one thing you see for these larger size class of particles, definitely wastewater treatment is working. This is shown across a lot of studies. I'll show you in the next slide as well. So you see a significantly higher concentration of plastic in this wastewater influent sample compared to the effluent. And we only have, at this point, maybe 5 to 10 stormwater samples in here, but we're continuing to look at those more, because you can see that our stormwater data is showing that it's looking higher than wastewater effluent. So I said we were not, you see that the wastewater effluent is pretty comparable to the levels that we're seeing out in surface water, albeit sometimes higher. You definitely see a significant decrease along wastewater treatment plant trains, and that's consistent with what's in the literature. This was a literature review we did, the studies of different particle sizes, and all the authors that did the hard work are there. And basically, every study was demonstrating log removal. How much is being removed varied across the studies, partly because their methods differed and the size classes they were looking at differed. But there's just a growing body of evidence saying that although there is some plastic left in wastewater effluent, the majority of it seems to be getting consolidated in biosolids, which certainly raises questions where there's land application of biosolids. Something that folks are interested in as well, I'm not a toxicologist, but I thought I'd just give some overview of the evidence, or here I'm questioning lack thereof, of ecological impacts. There's strong evidence that there's ingestion of microplastics in the environment, and as well as in lab studies. So I've seen reports that over 100 species have ingestion, which I think is in itself probably concerning, right? The potential risk that's associated with that, it seems to be falling into one would be from mechanical issues. I think we've all seen studies or pictures of animals whose guts were clogged with plastic. And a secondary concern could be whether there's some chemical risk. Here I'm giving you a citation for a paper that I think is great, speaking about the chemical risk here. The idea being that, these polymers, they're usually used in food grade. A lot of them are used in food grade materials. So the chemical risk from the polymer itself is unclear, I would say. But it's when you think about the additives and the plasticizers, that there might be more of a concern as to whether there's chemical risk. An example there might be, I think folks remember BPA. That might be something that could be posing a chemical risk. That isn't necessarily the polymer itself. But figuring out those additives is certainly trickier than saying whether it's just a plastic or not. And there's also been some folks that did great reviews of the toxicology studies that are out there. And here's one where they did a quality review of those. And basically one of the main outcomes was that they saw that there's a wide variation in the quality of those studies. So if you're interested in following up on those, or if that's a motivation for regulations or anything like that, it certainly deserves some consideration, the quality control in those studies. One risk that folks have been interested in when it came to microplastics is what's sorbed to it. Like, are we concerned about the toxic chemicals that sorb to plastics? Are you concerned about the fact that they found pathogens on plastic? So that was something we were interested in studying in the lab. So I thought I'd sprinkle in a little of our results here. We wanted to study this for a variety of microplastic types, as well as morphology. So here are some films and fragments. But we wanted to make sure that we did this with material controls. When you look at some of the cross-study quality control, quality assessment work that's been done, a major criticism is that not every study has the proper controls, whether that's blanks or matrix spikes or material controls. So for here, we used wood chips as a natural material control. Incubated those in wastewater influent or secondary effluent, did some disinfection, and looked for fecal indicators. So microbes of regulatory interest in water and wastewater. And just to give a little view of the results, comparing what we saw in the biofilm and the filtrate of those reactors on either one type of plastic or the natural particles. Basically, on the left here, you're seeing that the wood grew more total coliform in the biofilm than the plastic did. not necessarily surprising. It probably has a rougher surface structure. It might actually be providing some nutrients that facilitate or help with growth. But in the filtrate, there was no difference. And then if we did disinfection, whether it was on the biofilm was on wood or plastic, there was no difference, no significant decrease in the amount of fecal coliform we observed. Again, that's not necessarily surprising. We know biofilms are harder to disinfect, but we're still seeing the disinfectant working. Here is peracetic acid in the filtrate. So just pointing out that, yes, you can absolutely measure some harmful, concerning microbes on microplastics, but I think we should always be comparing it to other materials. Here, results, I think, suggest just that removing particles in general would be good from wastewater treatment to avoid bypassing disinfection. So hopefully I didn't go over on time. Here's some of the papers that I cited along the way from our lab. We have a variety of collaborators working on different things, some folks working on the biosolids, on the biological uptake, and things like that. And I'd definitely be happy to answer any questions that folks have.
Thank you, Nicole. We're right at 17 minutes. So we're actually on time for once. That's great. So we can go ahead and open it up to some questions. So you can go ahead and put it in the chat box, or don't be shy, turn on your camera, and we'd be happy to go through questions like that. While we wait for that, I actually have a couple questions. I took some notes here while you were going through that. I guess my first one is, you had mentioned microfibers. So when you were doing your sampling, we have both microplastics and microfibers. Did you find a specific percentage, higher percent of microfibers versus microplastics in any particular bodies of water?
Yeah, that's an interesting question. For a lot of our studies, we'll characterize microfibers when we run into them, but we have not been doing them quantitatively like we have for the particles. But I can say from the other literature that's out there, it looks like microfibers, particularly as you get to smaller sizes, are more prevalent. The concentration of them is higher in a lot of wastewater samples and even surface water samples from other data I've seen out there. The challenge is you need really good air systems and good controls. There also have been field work that I've seen from groups where they said, look at all these fibers, and then they ran their controls and they're like, okay, they're all from our clothes.
Okay, right, yeah, fair enough. And I guess a follow-up to that is on the microplastics front. I know that it's very hard to identify to exactly where it's coming from. You could break it down to a chemical structure and say, okay, it's either coming from PPE or polypropylene or et cetera. When you're testing it, for example, there's all this push for biodegradable plastics. And that you can identify because you can break down the structure and go all the way down to identify exactly where it's coming from. Do you see this push on biodegradable plastics as a higher indicator that we might have more microplastics out there? Like, will it be a main contributor, I guess?
I guess it depends on how they break down. If they're biodegradable in that you can actually break the polymer structure, then I don't know if they'd be resulting in smaller particles out there. Actually, I actually don't know as much as I should about those.
Okay. Yeah. I mean, they claim that they're biodegradable. The question would be, how biodegradable are they, and how do they break down, right?
And certainly the mechanism. That was something that came up at WEFTech last week. Is it biodegrading just that it's mechanically breaking down into something smaller like you expect with the photodegradation, or is it really biodegrading where you're getting the chemical structure breaking down?
Right. Okay, excellent. Well, those are my questions. I know I kind of promised everything you need to know on microplastics, and it's hard to do that in a very good and compact 20 minutes. So I hope we answered some of the questions. Thank you, everybody, for joining us. Thank you, Nicole, for being able to do this presentation. Again, I want to give a little reminder for next week. We're going to be touching on cyanotoxins and cyanobacteria, also known as algal blooms. We're doing that on Thursday at 10 o'clock. If you don't already follow us on LinkedIn, please do. We put up some of our updates on some of the webinars and some of the news that we're going through. I am going to put in a shameful plug. If you take a look at your chat box, the chat box in there is going to have the YouTube channel where you can see the recording. And we will be sending out the recording to everybody so you can have a look. With that, I'm going to look at that right on time, 10:20. So with that, thank you very much for joining us this week, and I will see you next week. Bye-bye.